talk about biomaterials. So that includes cells, tissues, organs. So hopefully one day this is the goal, right? To be able to print yourself a new heart whenever you want. But unfortunately we're not there yet. Most of the bioprinting that goes on is for medical testing, drug development. But this has provided a lot of cost savings and better results, faster results. So it's an ongoing process, the development of the new technology. But as you can see here, there's functionalized liver tissue, which has performed for well over 40 days. So this is possible. Obviously, why do we want to print biomaterial? Well, we're made of biomaterial. So if we can replace our organs, our tissues, when we get hurt, soldiers when they get wounded, when someone needs a new heart or whatever, that's obviously a good thing. So the chemical compatibility can be customized on a per patient basis at the genetic level, at the cellular level. All the things are really great when you're trying to put something foreign in somebody else's body. So some typical applications, like I said, they've printed liver cells. This is a aortic valve they recently printed. This is a cartilage they're printing here. This is an ear recently, so the ear was... A lot of the printing that happens now is they print a scaffold. They print the scaffold, they infiltrate it with cells, and cells, have this way of growing on their own when they're provided the right nutrients. So that's usually the way the printing is happening now. This is skin they've printed, and this is actually arteries. If you can see inside the petri dish, they just recently printed arteries, blood vessels, so epithelial tissue. Alright, so the bioprinting process, same as what you've seen before in 3D printing, but I just thought I'd go through it since this was a neat chart in this review article on bioprinting that just came out this year. So again, there's imaging, so you're going to look at the patient, see the structure that needs to be customized, so mechanically, but also then later you're going to look at the design approach and see chemically, what approach am I going to take? Am I going to try to mimic the actual chemical structure? Am I going to try to have these globules agglomerate by themselves? Am I going to create little forms of the cells and allow them to self-assemble later? Then is it going to be synthetic, like I talked about last time, biopolymers, natural polymers? Is it going to be some type of cell that's grown? Again, am I going to use stem cells? What am I targeting? Is it bone? Is it muscle? Is it tissue? Is it heart? So do I need stem cells there? Can I grow the cells themselves first? What type of method am I going to use? Right now, they actually have used what we know is inkjet-based 3D printing and applied it to bioprinting. Or, again, there's micro-extrusion. As you can see, they're printing a liver. Laser-assisted, which is pretty new. The papers just came out on this year. How do we direct the assembly of cells using a laser? And implantation, testing afterwards, and making sure that they're maturing correctly and growing. There's no toxicity compatibility issues. So as you can see, the inkjet bioprinter, thermal, piezoelectric based. Some of them are actually pneumatic based. I was reading. Then there's micro extrusion, same thing, screw or piston. And then the new one, laser assisted, which you wouldn't think, but if you tune to the right frequency, you can actually direct the growth, which is a kind of a key factor in growing bio tissue actually. So some of the material properties, importantly, this is different for all types of cells. So liver cells are going to be different. Bone tissues is going to be different, osteoblast. So here, the three main components are the structure has to be precise and consistent. They have to be viable, so they have to be fully functional, stable, so more than just a day. They have to stick around for a while. They have to be compatible, so you can't have toxicity, high toxicity. You have to be able to be compatible with the surrounding tissue. So then again, that goes, you're printing these cells so they can become this tissue, and maybe you're actually building up the tissue to form an organ or just replacing the tissue itself. So again, structure, viability, compatibility. So if we look at these osteoblasts, this picture right here, bone cells, there's 4 different types, so it's important again to choose which type. Then there's structure, so let's do some micrography and see whether the structure is okay. They look pretty great. What's the viability? So say for bone, the ultimate tensile strength we're looking for in our human body is 130 megapascals. So we want to make sure our printed tissue lines up to those parameters, the density, flux, blood flow, all types of things like that. Again, compatibility, looking for muscle and bone growth. Stem cells. So these are embryonic stem cells they've printed. Want to look at the ultrastructural characteristics, so what makes the overall matrix of these cells, what makes them work or organizationally, whether that's lining up, viability, and measures of self-renewal. So for stem cells especially, are they self-sustaining and can they stick around? Potency, compatibility, how efficient are they? If I want to direct them to differentiate into liver cells, can they do that? And can they do that better maybe than going to heart cells or something like that, or blood cells? So like any building block of any 3D printing, right? There's powder, which is a particle. Here, the building block is just like a blob of cells. So liver tissue, hepatocytes. So again, distinct cellular patterns is important. So ATP and albumin, which is a what is it again? It's a protein, and that really tells you the healthiness of how healthy the cells are. Right, so how well they're doing metabolism, how well they're using nutrients. Compatibility, so APAP is a type of toxic marker they look for. So biobots, you probably just heard the news, I did too. They're coming out with, what they're saying, they're doing the MakerBot strategy for bioprinting. So essentially making it very affordable and efficient and simple, so researchers can put this on their countertop, make 3D assemblies for drug testing, all other types of testing. So the printer is supposed to be $5,000, and the Bioink, so those starter packs, right, a pack of those is going to be $700. And there supposedly you can make bone, liver, heart, all types of cells, skin. So there are a lot of possibilities. It's inkjetting again. BioBots 1. So it's supposed to be the more affordable and efficient bioprinter. And again, so there's the curing here. It doesn't happen by UV. UV can damage and also dehydrate the cells, which doesn't promote their continual self-renewal or growth or viability. So there's a blue light, which in combination with some combining agents already in the Bioink, it allows the material to grow. So, all right, here's so they, at South by Southwest, they printed Van Gogh's ear. So there's that with this printer. All right, so my case study is by actually a company called Organovo. They work with industry and government. Again, right now, popular way, I think the only way of bioprinting is inkjet based. So they print tissue for drug development and medical testing. So it's not a layer as you would think in traditional 3D printing. A layer is not just a layer of cells. It's actually about a layer of 20 cells thick, and that forms each layer of the tissue they're trying to build. So it's beneficial because there's no scaffolding actually with this process Organovo has. They're able to achieve accuracy, like I was saying, which is important to make sure the cells are functioning well, so that they're biologically relevant. So that means that they claim that their tissue actually feels and looks like, to medical professionals, looks real. I don't know how accurate that is, but at least they say that. And again, it's customizability based on patient inputs, not inputs. But they have a lot of ability to say, hey, my body reacts in a certain way, so here's what we're going to do. So here's the printer. It's a pretty cool image from Wired, I think. So yeah, so I have some notes on how this works in case you're curious, like Ian was. So in step 1, engineers load a syringe with Bioink. They're made up of these spheres that contain tens of thousands of, in this case, liver cells, and a second syringe with Bioink containing another type of liver cells that are different, that are supposed to bolster development, and also a hydrogel that helps with extrusion. Then software commands the bioprinter and the motor to move and lower the pump head with the second syringe, which begins to print a mold. The mold prints a honeycomb pattern. The sensor tracks each tip along the axes, and based on the location, they determine where the first syringe should be positioned. The robotic arm lowers with the first syringe fills the honeycomb, the engineers remove the well plate, which contains the completed micro tissues. So it's a complicated process, but it's able to produce bioprinted tissue for testing, at least. That saved pharma companies a lot of money and time. Okay, so yeah, there's that tissue, and again, here's the bioprinter, just another shot of it on the lab. So as you can see, the albumin levels and ATP levels, which are a sign of viability, another measure of this material, are performing well in comparison with actual liver tissue and over a range of time, which is important as well. But if you see the albumin levels, they're not disrupted, and the ATP levels are increasing. So that's something you want to see, and you see in normal tissue, healthy liver tissue. So for my first research study, I looked at cell and organ printing 1, protein and cell printers. So bioprinting basically started back in 1997. Two biggest groups that worked on it, where one was from Clemson and one was from University of Missouri, Columbia. The first one from Clemson, they worked on the actual converting commercial inkjet-based 3D printing into some type of technology that could be used to make biomaterial. And the second group worked on actually making those spheroids, the type of bioink that's out there now. Basically, both these groups and these papers established bioprinting as it looks today, so I thought it'd be important to talk about the experiments that they did. So basically, they tried to develop a inkjet type 3D printing process for organic molecules. You can see the printer there. It's a poor lab image, but that's what was provided. It's a converted commercial inkjet. I think it was HP. And yeah, so they jimmy-rigged that thing, and you can see inside of it. So they fabricated samples of cells and proteins. So the first one you can see here is just protein. So they tried to, they had 4 proteins. They had BSA, biotin, BSA, the one with the S, and the Y biotin. And they're down there below if you're curious. And so the biotin, after they washed it with water, they didn't last. The only thing that lasted was that T in yellow that you could see. So you could see that they could print it, but the adhesion of the protein wasn't best. But still, after they did some residual scans, it was better than what they had thought. They hadn't wiped out so much of the proteins. And here they printed epithelial cells, so bovine epithelial cells, which are the cells that line your arteries. And here is particularly the ones that are leading into the heart. So the first one was an SA after it was printed, wasn't allowed to grow. B is after it's allowed to grow. It's a strip of smooth muscle cells. So again, they were able to convert commercial hardware and software into something that could print protein and, importantly, cells which are able to grow. And they didn't say if they performed as well, but I think the objective was just to be able to do it, since this was back in 2003. Second research study is engineering biological structures of prescribed shape using self-assembling multicellular systems. So this is a professor from Columbia, Missouri, and he's trying to see if these spherical globules of basically cells can be like these starter packs, or these things that you can inject into your substrate and they be allowed to grow. And so you can see in E there, they've printed this in this torrid shape that he said, the globules, this spheroids they call them. This has become the most popular bioink option, and they eventually fuse over time, as you can see, to become living tissue. So again, this was a kind of a foundational thing to, yeah. So as you can see, they contract inwards, right? The inside area shrinks, and that's actually an important indicator, the paper says, of if they're growing well or not. So another thing about the droplets here, the spheres, they're not just cells, right? They're also biogel. So this biogel, you can tune the chemistry and actually enable the growth, change the way it grows, faster, shorter. You can actually change the geometry. So that's what they found in this paper. So you can see here, A, B, K, and L are simulations. So they did computer simulations. They developed a model based on the energy of that surface tension between the spheres, and they actually used that to calculate the best angle and the best amount of time in order for this to actually grow. So you can see the top row becomes the bottom row, and these are for different angles. So this is E. So look at E here. This is what E becomes over 168 hours. So, he was wondering if you could use these things as self-assembly blocks, and, it ended up you can, and we do use it in Bioink. And so here you can see, again, over time, but you can see what I was talking about, the angle and the importance of this surface tension and adhesion, importantly. And over time, you can see that the angle goes from, as they plotted here, very low to almost a straight line here at 150. So it's pretty cool. Kind of set up what bioprinting looks like right now. And in the future, we're always headed to hopefully printing all sorts of neat things. But that's where it stands now. But